As they say that the glass is never half filled and half empty, but it has several genomes in it. And I also abide by the same. And if you also abide by the same, and if you also want to know about the same, you have come to the right place. So hello and welcome. This is Himanshu Kaushik, your host of the day. And I would like to welcome you all in today's webinar on understanding DNA and proteins, integrated bioinformatics approach. And I hope that you all are fine, safe, sound, and happy at wherever you're taking this webinar from. So before we move on and learn a lot more about the process or informatics process that we're gonna learn through the session today, we have our expert in here. So I would like to request Dr. Gurmeen ma'am. She's an assistant professor here. She kindly please introduce our expert with us and all the attendees, ma'am.
Thank you, ma'am. So in this online series of webinar, today we have Dr. Shikha Suman, Assistant Professor, Department of Bioinformatics, School of Bioengineering and Biosciences. She completed her Master's and PhD in Bioinformatics from Indian Institute of Information Technology, Allahabad. Her PhD research topic was identification of potential biomarkers for the early detection of cervical intraepithelial neoplasia. Her research areas include machine learning and artificial intelligence in bioinformatics, data analysis, next generation sequencing, RNA sequencing, and single sequence data analysis, biological network analysis, protein modeling, and simulation. Total impact factor to her credit is 18.87. And this is very recommendable. She has published her research findings in journals of repute like Scientific Reports, Oncology Letters, and Molecular Biosystems. She secured first position in Navdhara Award session in ISES 2019 and Max Cancer Congress held on 18th August 2019 in New Delhi for the most innovative research. She also received Professor Joel Cohen Tanauji Gold Medal 2014 for the first position in MTech at IIT Allahabad. She received Vice Chancellor Silver Medal for securing second position in BTech at the university level in 2011 at Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut, India. She also won poster making competition at RJIIT Amethi UP in 2012. She received the ICAR fellowship from 2007 to 2011 and the MHRD fellowship from 2012 to 2019. Today, she is the resource person of the day and she is going to deliver a talk on understanding DNA and proteins in integrated bioinformatics approach. So students, visualization of nucleic acid and protein enables researchers to visually explore and analyze the complex molecular structures and interactions. Bioinformatics provides tools and software for visualizing nucleic acids such as DNA and RNA in a three-dimensional space. Visualization techniques help researchers understand the structural characteristics of nucleic acids. By visualizing nucleic acids, researchers can analyze their conformational changes, identify binding sites, and study their interactions with proteins, drugs, and other ligands. Similarly, bioinformatics plays a vital role in visualizing proteins, which are complex three-dimensional structures with critical functions in biological systems. Visualization tools allow researchers to depict protein structures such as the arrangement of amino acids, secondary structures like helices, sheets, and tertiary structures, the overall fold. Bioinformatic tools also enable the visualization of protein ligand complexes, providing insights into drug binding and the design of novel therapeutics. Hence, by combining computational tools with visual representation, bioinformatics enhances our understanding of the molecular world and enables innovative approaches for solving biological problems. So, Today we are here to listen to a very good topic by Dr. Shikha Suman. So I would now request Dr. Shikha Suman to present her presentation and then the demonstration.
Okay, I would just like to take another minute, ma'am, before Shikha ma'am commences with the webinar. I'd like to inform all the attendees, all the participants, that you can post your questions in chat box, Q&A box, and we can allow you to talk if your questions are of worth, so that you can have a direct session with our experts in here. So now, ma'am, the stage is yours, and I'd like to request you to kindly please take over the screen, and looking forward for an amazing session, ma'am.
Thank you, Gurmeet ma'am, and thank you, Himanshi ma'am. So students, good afternoon all. Today we are here to learn something about DNA proteins and how we can visualize these DNA proteins, which are the building blocks of any organism, using computational tools, basically the bioinformatics approach. So you all must have known about DNA and proteins as in your basic study, you have must have studied about cell and how these cells are organized and it makes an organism. So cell, the collection of cells are called tissue. Tissue are then collected, group of tissues are called organs. Different organs club together to make organ system. There are different organ systems that come together to make an organism. The same process works for both animal as well as plant. So from cell to organism, we have different level of organization. So when we look inside the cell, there is nucleus, and inside the nucleus, there is some thread-like structure that is called as chromosome. So now chromosome is nothing but the winded thread-like structure, a simple thread that is winded so that it can be fitted into a small space. Okay. So when you say DNA, so what is DNA? DNA, RNA, these terms are very commonly used, and these are the most important building block of any organism. So what are these? These are simple nucleic acids. So nucleic acids are polymers that consists of nucleotide residues. Okay, so these resides inside the nucleus of the cell, and these are the hereditary determinants of any living cells. Why it is called hereditary determinants? Because the way any DNA is encoded, the way any nucleic acid is encoded, it decides how that organism will be, whether it in terms of its look, whether it in terms of its behavior, or whatever it produces. Everything is dependent on the encodes of that particular nucleic acid. So further we will study that how these nucleic acids are made up of, what are the constituents of this nucleic acid. So basically, any elemental compositions of any nucleic acid, whether it be DNA or RNA, the elemental compositions are carbon, hydrogen, oxygen, nitrogen, phosphorus. So broadly, when we say any nucleotide, it consists of three parts: phosphate group, sugar group, and base. Now, there are 4 types of nucleotides we have, and the combination of these 4 types, different combination makes different phenotypes, or how any organism looks, or how any organism behaves. So here you can see the bases. Bases are purines and pyrimidines. And in base purines, we have adenine and guanine, whereas in pyrimidines, we have cytosine, thymine, and uracil. Thymine is used in DNA, whereas instead of thymine, uracil is used in RNA. Otherwise, these 4 bases, combination of bases, makes different nucleotides. So when we talk about nucleic acid, we have deoxyribonucleic acid and ribonucleic acid. Deoxyribonucleic acid is double helical structure, the first diagram you can see. And the second structure, which is the single stranded. The DNA is double stranded, but the only single stranded, if we consider the only single strand of any ribonucleic acid, that will become RNA. So all this is, you can assume it as a thread, and in that thread, we have different nucleotides, and different nucleotides can have, we can say there are different nucleotides based on the different bases it has. For example, it has, the base is adenine, so we can say it's adenine nucleotide. If it has cytosine, we can say the nucleotide is cytosine. If it has thymine, we can say the nucleotide is thymine. And if it has guanine, we can say the nucleotide is guanine. Only difference between ribonucleic acid and deoxyribonucleic acid, that is DNA and RNA, is the presence of uracil instead of thymine. So you can see in the diagram also, instead of thymine, we have uracil in RNAs. So once we know that inside the cell, we have nucleus, and inside the nucleus, we have DNA or RNA. Basically, if you talk about human cells, so we have DNA. And DNA is just a thread-like structure where we have different nucleotides encoded. So how this different encodes decides what type of feature it will have. Every feature, for example, if I say the color of anyone's eye, color of hair, color of skin, or any feature is represented in terms of or in the form of proteins. So the DNA is converted into RNA, RNA is converted into protein to express itself. So if we say that the person is having certain codes of DNA, for example, in this diagram, you can see there is certain code mentioned, that is T, C, T, G, C, T, something like this. So what is this? This is the sequence of DNA with different nucleotides in it. So if this combination of nucleotide with the process of transcription makes RNA, where T is converted into uracil, because we have already studied that in RNA, instead of thymine, there is uracil. So T is replaced by U. And further, by the process of translation, the RNA will be converted into protein. Protein is the main expression factor through which any expression, for example, I have said that the color of the eye, if it is black, so there will be certain code of DNA that will code for certain RNA, and that will again code for certain proteins. If someone has brown eye, so the protein will be different. So hence corresponding RNA will be different, and corresponding the basic DNA sequence will be different. So what is proteins? Proteins can be any enzyme, any defense proteins, any transport proteins, any regulatory proteins, any structural proteins, movement proteins, and so on and so forth. So whatever features we can say, feature means what defines any organism. Whatever feature is there, that is due to certain kind of proteins. If I say enzymes, so enzymes are proteins that is used for catalysis of any reaction. For enzymes are important for any reaction to take place, whether that is catabolism, metabolism, or anabolic or catabolic reaction. So apart from that, we have defense proteins, for example, antibodies. So when we will study about viruses, so there are antibodies for that. So antibodies are again certain type of proteins only. They are hemoglobins, myoglobins, these are transport proteins that is used to transport iron from one cell to one place to another, or one cell to another cell. Again, we have regulatory proteins, structural proteins, keratin, which is the component of any hair or significant protein is this. Again, we have movement protein, actin or myosin. There are different types of proteins, and all these proteins are made by DNA, and with certain code will be there for different proteins. So proteins are nothing but the molecular machines, building blocks, and arms of the living cells. Because any skin, whether it be skin or any organism, it is made up of proteins, and these proteins are made up of amino acids. Amino acids are the building blocks of any proteins, because proteins are the polypeptide chains, and amino acid makes that polypeptide chains. So proteins are polymers. Polymers means it is made up of multiple amino acids. That's why we are saying these are polymers. Okay, so in any amino acid, the only difference will be the presence of different amino group. Otherwise, there will be some side chain, hydrogen group, and carboxyl group will be there. Amino group will be changed, and side chain will change, and it will ultimately change the type of amino acid. So 20 standard amino acids we have, and the combination of these amino acids makes thousands and lakhs. It can ultimately lead to different types of protein. So as we have studied in the form of DNA, there are only 4 nucleotides, and the combination of those 4 nucleotides, there can be infinite number of combinations. So how these nucleotides are combined, it makes different DNA sequence, and ultimately the protein will be different. Similarly, for amino acids, the combination of different amino acid, the arrangement of different amino acid in the sequence, that leads to different proteins. So when we see or we want to study the level of how any amino acid, that is the building block of any protein, is converted into or made into a full-fledged protein molecule, there are 4 stages. First stage is simple primary structure, where each amino acid are just connected to each other using in the form of simple polypeptide chain. Okay, using with the help of peptide bond. That will be simple thread-like structure, you can say, where different amino acids are there. Further, these simple thread-like structure is, we need to ultimately make a condensed form of protein so that the primary structure is folded to make it a comprised form. So we have alpha helix or beta sheath, where the helical structure is called alpha helix. The simple thread-like structure or simple sequence of amino acid, that was the primary structure, further binds together to make alpha helix. Another form of secondary structure is beta sheets or loops. Different forms of secondary structure are there, where simple thread of amino acid or sequence of amino acid is further concised. Then the secondary structure, different secondary structure, let's say I have said that there are different types of secondary structure. There are alpha helix, there are beta sheets. These different secondary structures are bring together, they are combined together to form the tertiary structure. That is the more complex or comprised form, concised form. Okay. And the last one is quaternary structure, where, for example, we have more than one polypeptide chain. So we can club those different primary structures or polypeptide chains to make a quaternary structure of any protein. So this finally quaternary structure of a protein is the main protein, and this is how any expression is there. With the help of any protein, we can say the expression will be, if I have said the color of the eye, so how this protein will be, finally, the expression will be in a simple such way. So I have said that there are basically simply, in brief, we can say that there are 2 types of secondary structure, alpha helix and beta sheet. So as you can see from the diagram, alpha helix is spiral. In simple spring you have in pens, no? So simple helical structure is this. Simple helical structure is called alpha helices. And when we have sheet kind of thing, simple sheet kind of arrangement is there, that is called beta pleated sheets. So we have alpha helix, we have beta sheets. These 2 are combined together to make tertiary structure. Tertiary structure, where when we have multiple tertiary structures, they are put together to make a quaternary structure, and that is our final protein. So understanding DNA and protein in simple terms, because now we are facing coronavirus. So virus is the simplest organism, you can say, in which we can say there are very few small strands. There can be RNA or there can be DNA. So how this virus looks? Here in the diagram, it is very nicely explained that you can assume a virus like a simple ball. Inside the ball, there is RNA. RNA, you can say RNA or DNA, we have studied as the form of thread. So you can say inside the ball that there is that RNA and DNA, and whatever sequence is there in that RNA or DNA, or in general, you can say nucleic acid, it will form some kind of, or it will express some kind of proteins. That particular protein is shown or expressed on the surface of that ball, you can say, and that virus is the combination of all these. So generally, what you can see, there are some DNA molecule or nucleic acid. In the terms of, for example, coronavirus, we have RNA. Then there are some envelope proteins. Envelope proteins means that ball we can say as the envelope, inside which we have the RNA. And then we have some spike proteins. Spike proteins that is present on that envelope. There are some lipid membranes. The envelope is made up of lipid membranes. So whenever you will have some kind of virus, there will be few proteins it will encode for, and all those proteins are expressed on the few important proteins are like spike proteins and lipid membranes. These makes the envelope of that virus. So in coronavirus also, we have RNA as the nucleic acid. Then we have spike proteins that is present on the envelope of that protein. Okay. And then we have hemagglutinin esterase. Hemagglutinin, this particular expressed protein is the antigen that is used for when we have some vaccine already we have taken. So how our body finds out that there is coronavirus in our body with the help of this hemagglutinin esterase only. This antigen acts as the antigen, and the antibody we have, because the vaccine once we have taken, the antibody is developed in our body. So that recognizes this hemagglutinin esterase and it captures this virus and destroys. Okay. So if we say that last 2 years and 3 years, we have been seeing there are different variants of any virus, this coronavirus. So we had Delta, we had Omicron. So there are different mutants of virus we have been seeing. So that mutants is nothing, but we have, because I told you earlier also, everything or every protein, any virus or any organism expressing, that is due to the sequence that is present on the nucleic acid. So if there will be any change, whether it be 1 change, 2 change, or any minor change will be there in any of the nucleotide. For example, here we can see we have normal sequence where that is made up of ATGC nucleotides, and we have mutation. Mutation instead of T, C has been incorporated. Mutation can be due to anything. There can be an environmental situation. There can be survival of the fittest. Due to that reason, sometimes organism change itself. So mutation means the change in the nucleic acid sequence. So here we can see T has been changed with cytosine. So once the T has been changed with cytosine, the type of protein this T sequence will change. And ultimately, for example, let's assume that the sequence here is for hemagglutinin antigen only. And now if this T has changed or mutated to C, the hemagglutinin antigen, the structure of it will get affected. It will change. So once if its structure will change, and I have a vaccine of virus related to Delta strain, and now it has mutated to Delta to Omicron. So my body will not be able to recognize those strains because those hemagglutinin antigen, because the structure has now changed. So how this structure works? Simple. There is lock and key method, you can say, where there is some antigen and there will be some antibodies. The structure of antigen is recognized by antibody and it binds to it. And there our body has the mechanism of destroying the antigens. So simple, when we have a virus, we have vaccine, we have vaccinated ourselves. So we have antibodies in our body. Once there is mutation, the structure of antigen will change. And whatever antibody we have, that will not be able to recognize those antigen, and we will again get infected with the Omicron virus. Okay, so mutation, if we are saying there are different strains of mutation and why it is again and again affecting human beings, because of this situation only, where even in the one change in the nucleotide sequence, the structure of the protein changes, and ultimately the antibody that we have is not able to bind with that antigen. So this was the basic protein and nucleotide concept I wanted to tell you. So using the bioinformatics, what we can do, because working in the laboratory is different thing and working in the computer to understand DNA and protein is different thing. So using bioinformatics approach, which is the computational method of handling or understanding or visualizing protein structures and nucleic acid structures or any biological component using computation, if you want to study that, that is called bioinformatics. So why it is important? Because before going to laboratory where human resource is available, different chemicals are involved, different price because chemicals are costly and time consuming. The experiments are time consuming. So different, these aspects are involved. So before implementing these aspects on the experiment, what we can do is we can perform the same experiment, or we can understand how that experiment will work on the computational level, on our system only, or our computers only. So here what we can do, virtually we can perform computer, or virtually we can see how these proteins or nucleotides are working. Okay, so here I'll show some of the tools, you can say, where we can, let's say I'll take some proteins and nucleotide as example and show you that we can visualize these proteins and nucleotides in different computer-based tools. And we can study these nucleic acids and proteins. So I'll share a screen of a tool that is called PyMol. And there I'll tell you how you can visualize different proteins and nucleic acid. Before that, I'll show you where we can get these proteins and nucleic acids, because we can create the sequence. If we know that certain nucleic acid or certain protein is made up of certain sequence. Sequence means if you are talking about nucleic acid, there will be ATGC component sequence. And if you are talking about amino acid or proteins, there will be the sequence or polypeptide chain of amino acids. And each amino acid is coded by certain letters, because I told you there are 20 amino acids. There will be 20 capital letters, English letters, symbols are there, and that can be used as sequence for any proteins. So I'll tell you where you can find out. For example, if I say I want to study about insulin, I want to study the effect of certain molecule or certain drugs on the insulin. So before going to laboratory, what I'll do, I will first see the interaction and how it interacts with how its effects on that particular protein or insulin, if I have certain drug. So where will I get this sequence? So first I'll show you different databases or platforms, that is online available platforms that you can just Google and find out. From there, I can find out different, whatever protein I want. I can download that protein. And then further, I can use different softwares. Here I'll show you how you can visualize your nucleic acid and proteins in PyMol, PYMOL. Simple. This is the simplest protein visualization tool. And you can play around with the different structures. So I'll share some of the tabs, some of the online sites. Okay, so if you can see, this is a site. I hope, yes. This is a site called Protein Data Bank. Okay, Protein Data Bank is a site which has all the proteins that have been searched and studied till date, all the information you can get from here. And from, for example, if I have searched for insulin, I can type here insulin, I'll get the suggestions of all the studies that have been there. So from this, I can download different, whatever file I want. I want the sequence, because I told you any protein has primary structure, secondary structure, tertiary structure, and quaternary structure. So for example, I just want to download the sequence of that protein. So I can just download it from this site. Okay. So similarly, we have different sites for, let's say, I want to download nucleic acid. Okay. So you have studied about NCBI. You must have heard about NCBI. So NCBI is Biotechnology Information Site, where you can download your protein of interest or nucleic acid of interest. I'll share the link. I think mine is hanged.
Ma'am, just to inform, your screen is not visible.
I think it's hanged. I'll just take 1 or 2 minutes.
Okay, no issues, no issues.
As I told you, we can visualize our DNA or proteins or any nucleic acid with the help of the tool called PyMol. So PyMol, here what we can raster file as I have demonstrated right now. So you can download the sequence or structure, and then we can, using the help file menu, we will go to open, and wherever we have downloaded the file, we will select that file. Here I want to show you any nucleotide sequence. So I'll open it, and this is the file I have downloaded, and I want to visualize and study this file. So when I had a sequence of nucleic acid, which I wanted to visualize, I opened it in the PyMol, and here I have started visualizing it. Okay, so there are different functionalities we can do with this structure. Just by seeing this structure, we can say that this is the nucleic acid only. And since it is double helical structure, we can say this, there are 2 strands, and you can play around with this sequence structure also. So this is double stranded. So we can say this is DNA, and this is double helical structure. So if I want to play with different presentation patterns are there. So what we can do, we can choose different looks for this particular protein. For example, I want to show it in the form of cartoon, or I can want to show it in the form of dots. Okay, so, and using this hide tool, we can change the conformation of these. Okay, there are different color options are also there. If you want to highlight single color, make your particular DNA of single color or multiple, you want to play around with that, that also you can do. Okay, so similarly, if we want to study about different proteins, so for example, I have a protein, this one. Okay, so I have downloaded sequence from PDB, amino acid sequence, and then I want to visualize the structure of this protein. So using this PyMol, I have imported the structure, and now I want to visualize it. I want to visualize this particular protein in different formats. Okay, so if I want to just see it in the line format, so there is line option. If I want to see it in cartoon format, and I'll hide the line option. So this, just by seeing this, I can say that this protein is made up of both beta sheet as well as alpha helix, because we have already studied that protein, there are secondary structure of proteins. It can be alpha helix, beta sheet, and the combination of alpha helix, beta sheets, and these are the loops. These combination makes the tertiary structure, and where we will have multiple tertiary structure, we can say the protein has the quaternary structure. So in this protein, we can see which amino acid or which section has double helical helix, alpha helix, and which has beta sheet, and how these loops are used to connect between different secondary structures. So just by using these options, we can see the compositions or different secondary structures of any amino acid or any proteins. So how we have used this? We can go to file, then we can use open. Then, for example, I have another protein molecule, and this protein molecule, the name of this protein molecule in PDB, the site I have shown you before this PyMol, the 4-letter code, for example, here the different nucleic acid or proteins are represented by 4-letter code. So in PDB, all the entries for whether it be protein, DNA, or anything. Everything is given 4-letter code as its identity or accession number. So here the accession number is 7BZ5. So here you can see when I have imported another protein, there is previous one also. So if you want to hide this previous one, so what you will do, you will go to hide and hide everything. So now I can see the only 7BZ5 as the complex protein, because this is the big protein where there is different beta sheets are there, alpha helix are there, loops are there, and there are, I think, multiple tertiary structures are there that has been clubbed together to make this complex protein. Okay, so you can move it and see, understand its interaction and all. So if you want to see the interaction between different proteins, so how you can do that? For example, and when you want to see or club together different proteins and let's say nucleotide. So here we have 2-KNL as nucleotide, the first DNA molecule we have seen. So we can club them also. Okay, so it will come in between this. So this is the way how any protein is visualized. There are other information or other features of this site also. Apart from PyMol, there are different tools, there are Discovery Studio, there are different visual tools that you can use to visualize your protein. So apart from this, what would I like to explain you is just the way we studied PDB, Protein Data Bank for proteins. There are different databases. The most important one, that is NCBI. I would like to make you just go through this, how and we can download the sequence where we can see those sequence so that further we can analyze it. So I'll share a screen for NCBI now. Okay, so this is the NCBI site where I have searched for insulin protein and I have got different suggestions where I have chosen IRS1, that is insulin receptor substrate. Okay, so when we go through this page, I'll get all the information about insulin, because nowadays there are n number of work going on each and every protein. So where all these information are stored, because I am a researcher and some other researcher is also doing the same work, then there is no point in repeating the work. So to save time and for help also, what we can do, whatever work has been already done and has been already verified, we can get it from different sites, different databases, and we can use that for further study so that our time is saved and also different resources are saved. So there are different databases. The most important one, I told you, this is NCBI, where you can get information about everything. For example, there is certain gene, there is certain protein, and its information is available. There are different experiments which has been already verified. So that information we can get here. So when I type anything, for example, I'm typing insulin. So what I'll get here, I'll get all the information related to it. Whatever experiments are there, whatever, all the genes related to it, all the proteins related to it, and so on and so forth. So for example, this particular protein, if I want to look at it, when I scroll down, I'll find it's all the descriptions are there. So how this protein is encoded, why this is important, what it encodes for, and different things. Further, when you scroll down, you will find, just I told you, any protein is made up of the sequence of DNA. So that particular sequence you can get from this particular site. So sequences are represented in certain format because it is a database, and in database collection of similar type of information, and similar type of information are always stored in similar formats. So here in this database, the sequences are stored in FASTA format. You must have heard of this term, FASTA format. So this is the simplest form of any sequence format. So here we can go to FASTA and download or see how our protein looks like. Okay, it will have some sequences. For example, when I see this, it has the sequence of nucleotides, different nucleotides, combination of nucleotides and there, and this is the sequence. Okay, so this sequence is the nucleotide sequence, because just by seeing the sequence, I can say this is made up of GC, ATGC. The component of this or the composition of this particular sequence is ATGC. So I can retrieve this sequence and then further work on it. Similarly, if I scroll down, I'll find additional informations also. So these are the database. I'll find this protein sequence. I'll find the different structures. Okay. So there are another databases also. For example, there is UniProt. So if I have certain protein and I want to study or I want to see what all works have already been done on that protein. So what we will do, we can go to UniProt. There just I'll simply type insulin. Let's say I want to look what are the different information that are available on insulin. I'll go to UniProt, I'll type insulin, and then I'll find its structure, its function. It is involved in different diseases. So what are the disease, what are the pathways, and different, whatever information that has already been studied till date, all the information we can get from there. We can retrieve those information and use it for further study, for finding any drugs, for finding any therapeutic diagnostic purposes, and many other researches also. So as I told you, we have UniProt, one of the most important sites for proteins. So this is how it looks. This is the front page or main page of UniProt site. So here you can see or you can find before going, as I told you, before going for experimental purpose, what you can do, you can study proteins or DNA at computational level. So here, there is search bar at the top of the page. There you can type your protein of interest and you will find all the information here. For example, here I have typed insulin and I am getting this particular information. Where right-hand side, you can see the amino acid. It is written 110. So this particular insulin protein is made up of 110 amino acid. Similarly, there will be left-hand side, you can see there are different parameters or different features we can retrieve or information we can retrieve from this UniProt site for particular protein insulin. If I click on name and taxonomy, so different names, different site-wise names, because for any gene or proteins, there are different names given to that particular. If you look for insulin, if I go to NCBI, I'll find different ID. If I go to UniProt, I'll find different ID. And if I go to PDB, the 3 database we have seen right now, we have seen that there are 3 different IDs given to this particular insulin protein at 3 different databases. These are called the unique identity number or XSN number. Then if I want to see disease and variants, means how particular this insulin is involved in disease, and what are the variants? Just now we have studied about coronavirus. There were Delta strain and Omicron strain. So those were the variants of certain strain. Variants mean there were very minute change in the nucleotide and the strain were changed. So similarly, we can find variants of insulin in this particular page. I want to see what is the structure of this protein. So I have, if already the structure is known, I can get this structure from this particular site. Here also, I can move it around and then I can visualize it properly. Just by seeing this, I can say that this insulin protein is made up of alpha helix, spring-like structure, and there are some loops. Just by seeing this, I cannot find any beta sheet in it. So this is the combination of alpha helix and loop-like structure. So this is again the simple proteins, we can say. Again, other informations also, because what are the processing, how these proteins are getting changed, all these things we can find from these databases. So this way we have studied about basics of nucleotides and proteins, and how we can visualize those nucleotides and proteins using some computational methods, some softwares, from where we can get the information about these nucleotides and proteins, and whatever research has already been done, the information that are already present on those nucleotides and proteins that we can get from different biological databases, how we can access them, how we can retrieve sequences from them. These things we have studied. So if you have any question, you can go ahead and ask.
First of all, ma'am, thank you so much. Before we go on to the questions, I'd like to thank you for coming in and for giving in your experience and for presenting this valuable presentation to all the students. And I hope that all the students must have enjoyed the session like I have. I've been listening to all the DNA and proteins, and I was getting all the flashbacks of my studies when I had done in 12th and 10th grade. So thank you, ma'am. And Kajal also says, thank you so much. So people, all the attendees are thanking you. And yes, ma'am, we have certain questions. So with your permission, can I take them up?
Yeah, sure.
Okay. Ma'am, there's a question from Janki, and she says, can we use Rasmol for protein visualization as well?
Yes, that is again one of the tools we can use for visualization. There are a number of tools. Pymol, I have showed you one of the tools, but there are a number of tools. Rasmol is again one of the good tools for visualization.
Okay, thank you, ma'am. Second question came from Radha and says, are these software free to use, or do we have to pay any penny for this?
Pymol is again, you can for 1 month, you can use it freely. Then if you're a student, you can make different accounts, and then after 1 month, you will have to pay for it. There are other tools that are freely available, you can find, but there will have some certain, when it is free, there will have lesser features to explore. And again, 1 month is there for working, and then after that, you will have to pay for it.
Okay, so student, you must have got the answer, Radha, that you can use free for a month, and then after that you can tackle the situation by either making different IDs, or you can pay. Okay, ma'am, we have another question from Ishani Hilal, and she says, do we need to go in bioinformatics here, or can we learn all these software while doing different degree, as in botany or zoology?
So pardon, what's the question?
So basically, she says that these software, she wants to learn these softwares, and she's not in bioinformatics right now. And she's that's what she's asking, that if she can learn all these things whilst being in botany or zoology.
Okay. Yes, of course you can use, because as I told you, whether it's botany, zoology, biotech, bioscience, in biology related subject, when you go for experiment, this bioinformatics approach or computational approach helps you save your time, money, and resources. So this you can use in wherever field you want. If you are doing botany also, so I told you the organization of cell is same, whether it's animal or plant. So you can use that. And these techniques are the basic bioinformatics techniques. Bioinformatics is not only about these things, but this is the one of the part of bioinformatics. So of course you can learn it from there, but when you full-fledged understand bioinformatics, then there are different things also. Nowadays we have expression analysis. You must have heard about NGS, next generation sequencing. Then there are single cell analysis. There are high level, high throughput sequencing, and its analysis are there. So that also comes under bioinformatics part only. But everything, and ultimately what we do, we try to understand how our cell works. Because for example, if I consider any disease, let's say cancer. So cancer is not because of dysregulation or change in one cell or one protein. That is due to the cumulative action of multiple proteins. So
Correct.
that we can, instead of going to laboratory at the first go, what we can do, we can understand this particular behavior or interaction in the computational level. So whenever you study biological thing at computational level, that becomes bioinformatics. But the basic step or the preliminary step of any bioinformatics or any biology related thing is by understanding proteins and nucleic acid. So here we have studied where we can retrieve these sequences, how you can start from basic, just visualize it and then work on it. So otherwise, when you go for bioinformatics study, full-fledged course and all, then you can study the higher level of it, the interactions, how different proteins can be studied together, and so on and so forth.
Thank you, ma'am. Ma'am, we have another question coming from Aditi Sharma, and first she is thanking you for the amazing session that you've done. And the question read is, are there any courses available to learn bioinformatics? Is Python necessary to work in the field?
I told you Python is the current language that is being used for understanding or implementation in that is being used in the bioinformatics nowadays. So Python is not required when you go for bioinformatics study, but since Python is considered as the easiest language till date, and it can be learned very easily, and it has multiple applications and implementations. So it is not required, but when you go for dedicated bioinformatics study, so you should, if you want to do better in this course, so you should go and learn this subject. So beforehand, you don't need to understand and learn all the Python and then go for bioinformatics study. No, you start studying bioinformatics, and side by side, you can, as the implementation application will be there, you can learn and implement. So nothing is too hard, but there are different colleges. Our LPU is also, there is multiple subjects related to bioinformatics and machine learning and Python and those kind of things. So there is full-fledged course of bioinformatics are there.
Yeah. So Aditi, I hope, yeah, so Aditi says thank you. So I think she has got the answer. Ma'am, we have another question from Saubhagya Day. And Saubhagya wants to ask about BLAST. And yeah.
So BLAST is basic local alignment search tool. So what is this tool? As I told you, when I showed you that UniProt database. So at the top left-hand side, you must have seen there is a tab called BLAST. Apart from that, when you search on Google BLAST in capitals, you will find the database also. So what does it do? Basic local alignment search tool means there is a term called as alignment. And alignment means simple. This is simple English term alignment, which aligns different things. So aligns means if there are multiple things and those things are similar to each other, it will align so that it can find the regions that are similar to each other. So why it is required? For example, I took one protein for coronavirus, or I have taken a sequence for coronavirus. There is coronavirus and it has certain RNA sequence. I want to find out a particular vaccine for it. And the time is less because making any vaccine or any drug, it takes years, 20 years. So what I'll do, I'll try to find out similar kind of virus. Are there any virus that are similar to this coronavirus? If there are some virus already present that is similar to coronavirus, if I find that and there are some drugs that are available for that virus, I can surely use that drug for coronavirus also. So what does BLAST does? It finds the similar type of sequence what I am giving the entry as. For example, I go to BLAST, there I am putting the coronavirus sequence, and then I want to search for all the similar type of viruses that are already present. So from that, I'll see what are the similar sequences. It will give me the list of sequences with percentage identity. There will be 90% identity, 100% identity, and so on and so forth. The list will be in the form of decreasing order. You can choose the top 5 or top 10 based on your purpose. And then you can also create the phylogenetic tree. Phylogenetic tree means how all the sequences are related to each other. Okay. if I say the relationship between human and monkeys are there. So how we can study that? Using this BLAST, we can study. If I'll put the certain sequence, human sequence in BLAST, and I'll find the similar sequence, I'll find some sequences or the same type of proteins in monkey also. So I can go for making the dendrogram of phylogenetic tree. Evolutionary relationship we can find out how these proteins or sequences are evolutionarily related with different organisms or different proteins. So those study can be done using BLAST. BLAST is the most important, one of the most important part of any bioinformatics study.
I definitely agree, ma'am. And with this, I would like to thank you again for resolving queries of our students, for giving in your time, and a special vote of thanks for the explanation that you've given whilst in the presentation, and of course, of the questions that student has asked now. So, and now I'd like to take your permission to show students about the admission process as well, because we've been getting a lot of queries about admissions, like how they can take admission. So I'd like to show them a little about LPU University site, so that they know that they need to go and what they need to do. So
Sure.
can I do this, ma'am?
Sure.
Okay. Okay. So to all the students who've had questions regarding the admission process, I just in a moment. Okay. So I think my screen is already visible, right? So what you need to do is, this is one shot resolution for every query of the admission that you have, that is lpu.in. So you simply post or you simply write this in the browser that you have, and after that this will be the interface that will be coming in front of you. Now, since we're catering about admission queries, we simply click on this admission here, and after that you can see that there are segregated programs that are given after 12th, after graduation, after post graduation, after 10th, doctoral, and they're very beautifully adjusted in here. So let's say we've taken an example of a student who has done his 12th grade and wants to take part in an undergraduate program. So simply go to after 12th, undergraduate, and regular programs. So once you click on regular programs, there will be a whole lot of disciplines that would be visible, as you can see in here. These are the disciplines that LPU has to provide to all the students. Now, you name any of the disciplines. So I'll be just maybe giving you an example: engineering, computer science, management, interaction design, public administration, and you name any of the discipline that you want to, and we have in there. So now let's take an example of because we were getting a lot of queries about the sciences specifically. So I'll be just taking okay, where is sciences? Yeah, here. So once we click on the science, we have different stream that are coming in under that, right? So simply click on any of the stream that you want to. So let's say I click on zoology in here, and once I click on zoology, we have a BSc honors zoology. We click on the particular course, and then we can see all the details in here. So program details gives you your eligibility criteria, right? You can see in here, this is the eligibility criteria. Highlights actually give you the student achievement, what all placements that our students are getting, or what all research that has been done. Fee structure would be, once you click on the fee, this will be a fee structure, but this is not what a student generally pay. Why? Because they get a lot of scholarships in here, and we have 3 categories of scholarships, and based on that you can avail up to 28% of scholarship on your program fee. And there are other scholarships as well. So you simply click on scholarship, and there are other scholarships. You need to explore and check whether you're able to apply for that or not, and if yes, then simply apply for the scholarship. Now is the important date, and that is 31st May is your last date of the admission. I hope that you've taken admission, or you have either taken the test. If not, then kindly go and apply, because it's not too far now, just 2-3 days now. And you simply, how you need to apply is simply click on apply now, and there are requirements that is asked from you, and you click on that. You'll be given your dashboard, and after that you will be getting an option to apply for the test or the course. And I hope that I've given you a basic idea as to how you can take part, how you can go to the site, and what you need to do. Now if I could not address anything that is there in the webinar, you can just post in Q&A box, okay? Or you can schedule a call with us. You can have WhatsApp, you just WhatsApp us, and we'll be there resolving your queries. or you can have live video counseling sessions as well. Now, last but not the least are the numbers of our university. So these are the numbers, you can call on them. Counselors, esteemed counselors will be here to provide their inputs about your queries and would resolve. So I hope that I've given you all the details as to how you can take part in, or how you can take admission in your course. And we have a question: is there graduate scholarships for international students at LPU? Yes, Ibrahim, there will be scholarships. What you need to do is you need to simply call or maybe mail us or WhatsApp us, so that our counselors will be providing you our apt answer of that particular question. So I believe I've made clear as to how you can take admission. Now, before we wind up and wrap this webinar up, I'd like to show you this one site, or not show you, actually, I like to post this site on the chat box. This is our webinar page, students. There are multiple webinars that are coming in every week, every day that we have. So you can simply explore this particular link and check which webinar you're interested in. It is all free. You can simply register there and try to gain as much as knowledge as you can from the webinars. Now, with this, I'd like to take permission from all the attendees, of course, and from the experts here to wrap this webinar up. If Gurmeen ma'am, Shikha ma'am, if you do not want to add anything to the webinar.
Yes, ma'am. Thank you. Thank you, Dr. Shikha Suman, for such an enlightening talk. And it was a very good demonstration of understanding DNA and protein structure using computational tools. Truly, ma'am, it was really exciting, since you have correlated it well in your PPT with the vexatious period of COVID. And this webinar really speaks volume of the expertise that you are having in artificial intelligence and machine learning. And this session also bears attestation to the fact that bioinformatics is truly changing the way research is being carried out these days, using numerous in silico tools. And I would not be wrong if I say that no research work is considered complete these days without the bioinformatics element into that. Yes, combining tools with the visual approaches help us to go deep down into the molecular level and understand their structure and correlating it with their functions. Thank you, ma'am, and thank you, Himanshi ma'am, for hosting this webinar. And special thanks to all the attendees who were there listening to the talk very with full curiosity, and their questions, whatever they had got, like, it really was commendable.
Yeah.
Thank you, ma'am.
Ma'am, pleasure is ours, ma'am, to be able to host you. And ma'am, Shikha ma'am, do you want to add anything to the webinar? Okay. So with this, I'd like to thank all attendees, of course, as ma'am has already thanked you, to be here in the webinar, to join in and to be so patient with us for this long hour. And all your questions, as ma'am has said, has been commendable and appreciated. Now, I would want to take your leave now, and until next webinar, stay safe and stay healthy, and kindly explore the site and other webinars as well, so that you can attend other webinars too. So with this, thank you so much, ma'am, joining in. Thank you so much to all the attendees for joining in.
Thank you.